Στην επόμενη ομιλία θα καλέσουμε την καθηγήτρια, την κυρία Amal Kaddoumi, η οποία θα μας μιλήσει για την έρευνά της πάνω στο θέμα των συστατικών του ελαιολάδου και την νόσο Alzheimer. Amal, Professor Kaddoumi, we welcome you to Greece, and we are so happy to be with us here today, and we are so interested to your talk and listen about your news, your new results about the activity of the phenolic compounds and the olive oil in general for the Alzheimer disease.
You have by the mouse? Okay. Okay, it's great seeing you everyone again, and thank you very much for this invitation, and I will be very happy to share with you everyone the findings from my research work, and I will prepare my presentation to show you the different kind of results that we obtained from different studies, so how we are moving from animal studies and reaching into human studies, and that's why what we call it as bench to bedside. So just here, this slide, just to show you the different hallmarks of Alzheimer disease, the pathological hallmarks of Alzheimer disease, where you can see here the beta amyloid plaques, you have the neurofibrillary tangles, neuroinflammation, oxidative stress, and also the cerebrovascular dysfunction, and that all collectively can result in a neurodegeneration and eventually can result into memory dysfunction and dementia. So just keep this in mind, which means here that we are talking about different pathological hallmarks that we are looking to target using extra virgin olive oil, using oleocanthal, using other phenolic compounds like oleuropein and oleuropein aglycone in order to control and hopefully that one day we can prevent Alzheimer disease with the right diet. So in the first study, when we talk about dietary supplements, we always talk about, and even in animal studies, we always talk about 10 milligram, 5 milligram, 20 milligram per kilogram doses. So the question of this study that we wanted to compare, when we take olive oil with very little compounds or phenolic compounds in the olive oil. So the idea of this study, we wanted to compare between olive oil that contains little of oleocanthal compared to that with olive oil containing or rich in oleocanthal, how it will affect the pathological hallmarks of Alzheimer disease like beta amyloid and related pathologies in a mouse model for Alzheimer disease. So in this study, what we did, we used animal disease model for Alzheimer disease mainly. It is known as 5-XFAD. And this is characterized by high level of beta amyloid accumulation. So for this study, what we did, we divided the animals to two groups. One group received refined olive oil. Another group received olive oil that is enriched with phenolic content or contains phenolic content, except oleocanthal. And the third group contains oleocanthal, refined olive oil spiked with oleocanthal only. And here, just to keep in mind that the dose we administered here of the phenolic content is only 0.5 milligram per kilogram. We added it to the mice diet for 3 days. So every day they received 0.5 milligram per kg. And when we talk about 0.5 milligram per kilogram, this translates into 75 micrograms per ml, per body weight. So 75 microgram per mouse body weight. So what we did, we wanted to see how it will affect the different hallmarks of Alzheimer disease. So what we found for this, both groups, that is low in oleocanthal but rich in phenolic content, and that group received oleocanthal in olive oil, we found that actually both of these groups compared to the placebo group or refined olive oil, they were able to enhance the cerebrovascular function or what we call the blood-brain barrier functionality by improving different protein levels, these proteins are key proteins necessary for beta amyloid production as well as beta amyloid clearance, which suggests that collectively can result in reduction in beta amyloid levels. So if you see here, the brown color here in all these figures represent the refined olive oil, and this one here is for the olive oil that is with phenolic content low in oleocanthal, and for this one here, this color here, it is representative for the olive oil that is spiked with oleocanthal. So collectively from this result, we find that both groups compared to the refined olive oil was able to reduce beta amyloid in mice brains. Also, not only that, we found that both groups actually were able to reduce inflammation or neuroinflammation in the brains of these mice. And we found that there are different mechanisms responsible for this effect via reducing the NLRP3 inflammasome activation, and also by reducing NF-kappa B activation pathway. However, interestingly, the oleocanthal group here, as you can see it here, it has an additional mechanism that the OC low olive oil group didn't show, which is called as RAGE HMGB1 pathway, which was specific for oleocanthal in this case, which provide additional mechanism for oleocanthal. So just to show you a conclusion from this study that compared to the refined olive oil at equivalent doses as low as 0.5 milligram per kilogram, OC low olive oil and OC rich olive oil effectively reduced beta amyloid and related pathology. And also both olive oil and OC, olive oil here means the OC low, olive oil and oleocanthal spiked olive oil, they reduced the neuroinflammation by targeting the NLRP3 inflammasome and NF-kappa B. However, if we want to compare, oleocanthal group was the only group that showed effect on suppressing another inflammatory pathway that is associated with Alzheimer's disease that is known as RAGE HMGB1 pathway, and all these are neuroinflammation pathway. So the idea here, maybe the take-home message that, yes, oleocanthal certainly is important, can provide additional beneficial effects compared to other phenolics, but at the same time, we should not also ignore the other olive oil that also contain good level of phenolic contents. Now, the other study we did here that we wanted to see how oleocanthal can attenuate amyloid-related pathology in Alzheimer's disease mouse model. So the objective of this study, again, we wanted to study the dose-dependent effect of oleocanthal on brain pathology and also on general toxicity in the body. So we tested this 5xFAD mouse model, we divided them, we gave them either placebo or 0 oleocanthal, 0.5, 1, 2.5, 5, 10, and 20 milligram per kilogram. Interestingly, we found as low as 0.5 milligram was able to reduce beta amyloid levels in these mice, and it was kind of, and interestingly here, for those who are interested, it showed kind of I will call it reversed U-shape, where at 2.5 milligram, kind of the effect was less than the lower doses and was higher than the later doses, which was an observation that was interesting. And we have a hypothesis for that, but I will not go through it now. So idea here that beta amyloid was reduced in a dose-dependent manner. This is the toxicity study. So this one here, I am showing only now 0, 0.5, 1, 2.5, 5, 10 milligram per kg doses. They were not toxic. The idea here, we gave 10 the doses were provided daily for 3 months. At the age of, for this study, it was from 6 months up to 9 months of age for these mice. And the idea here, the main result from this study, that at 20 milligram per kg, when administered to these mice, actually it resulted in some kind of hepatotoxicity. So while oleocanthal is good, diet, but we have to make sure that we do not exceed a level that with time can result in hepatotoxicity. And actually, when we talk about 20 milligram per kg is really a high dose if you translate it into human dose. So for sure, whatever we are giving subjects or patients, it will never reach this level, but for us, we wanted to understand the toxicity profile. Next, now we have to remember that when we talk about Alzheimer's disease, it is not only about the pathology, about beta amyloid, about tau, about neuroinflammation. Alzheimer's patients are usually depressed, they lose weight, they are less energetic, they don't sleep well. So the question of this study was, okay, we want to understand how oleucanthal will affect all these parameters if we are talking about Alzheimer's disease. So what we did here, I selected one dose here. We did it for different doses, but I selected the 10 milligram dose. We gave it for young mice. And at 1 month up to 4 months, 3 months treatment. And for older mice, because we wanted to see at early Alzheimer and at a later Alzheimer when it is advanced. So with the other group started 6 months up to 9 months. So we continued this for 3 months with oleucanthal with 10 milligram per kg into Alzheimer's disease model. We compared it to the Alzheimer's disease model with the placebo, and also we compared it to the wild type, which is cognitively normal function. And for all these parameters we wanted to study, we used a metabolic cages or a system called the prothrombin system that can monitor anxiety, sleep pattern, body weight, food intake, and all other things. And also we did some pathological studies as well on Alzheimer's disease hallmarks. Busy slide, but what I want you to take from this. Now, for mice, we divided the 24 hours into day time and night time. Daytime, which means in the morning up to noon, and then the night time is when at the night time. So what we found here, just for you, the blue color is for the normally cognitively function, which is the wild type. This is for placebo, Alzheimer model, and this is with the green or dark green is for the oleucanthal treated mice. As you can see here, we said body weight is decreasing with Alzheimer. So we found actually oleucanthal the effect was not significant, but it was able to increase the body weight to approaching that of the normally cognitive mice. Also, usually with Alzheimer, you will find that there are differences in energy expenditure in the body with Alzheimer. So we found actually in most situations, we found that oleucanthal restored these parameters to the normal level. Not only that, we found also that oleucanthal was able to reduce anxiety in these mice to levels that are, to levels that are comparable to that of the wild mice. So that was interesting for us, but the more interesting study results is the sleep pattern. Oleucanthal was able to increase the sleep pattern of these mice it was significantly different between the wild type. However, when we gave them oleucanthal, we can see that their sleep number of hours actually went up to levels that are consistent with those of the blue color here, which is for the wild type mice. And this is very important for you guys that in Alzheimer patients, sleep is lost. So oleucanthal, olive oil, honestly can restore that level into the normal levels. Now, from this slide, a lot of information there. I can explain some, and I cannot explain the others. However, here, because we are comparing young to aged Alzheimer's disease mouse model with oleucanthal and with wild type, the idea here, if we look at C and D here, C and D are for the beta amyloid levels in the brain. As we expect, it reduced it significantly. Interestingly, in the plasma, the effect was different at early age to the latest age or the advanced Alzheimer. While you will see at the young mice, it reduced beta amyloid, which is expected, like the brain. However, at the aged mice or with the advanced pathology, we found also that it restored it to the wild type level or the normally cognitively normal, but in the other direction, which is increased that. So I can explain some of this data, but not everything. So that was really interesting finding that we have from this study. Now, another study that we did in my lab, we wanted to study the oleuropein. So what we did here, it is oleuropein-rich olive leaf extract, and we wanted to see its effect on amyloid pathology in Alzheimer's disease mouse model, and I abbreviated here as OLI. And for this oleuropein-rich olive leaf extract, so it was olive oil, refined olive oil, and it is spiked with the oleuropein-rich olive leaf extract. That was the idea of the producer, but I think it is very also indicative of some effect that we could get. So for this study again, we treated mice at the age of 3 months to 6 months with oleuropein spiked or oleuropein extract spiked into olive oil, and we compared it to refined. Vehicle here is refined olive oil, or the olive oil that is not spiked with the oleuropein leaf extract. And we, for 3 months, and at the same dose of 0.5 milligram per kilogram, which means these mice received 75 microgram per day per body weight of the phenolic content. We can see here, consistent with the oleucanthal result, beta amyloid was significantly reduced. Soluble beta amyloid was also reduced, mainly A beta 40 for the beta amyloid isoforms. And also we found that oleuropein-rich leaf extract, we found that it also reduced in neuroinflammation, which we expected so, and there were different mechanisms where it reduced astrogliosis via measuring GFAP. It reduced different cytokines that their increased level is associated with Alzheimer's disease that was able to significantly reduce it compared to the vehicle or to the refined olive oil group. Not only that, we found actually that it enhanced the synaptic markers. Why the synaptic markers are important? Because they are related to the memory. And we found that oleuropein-rich extract rich in the olive oil significantly increased memory to levels that are similar to the normally cognitive mice compared to the mouse here. Compared to the vehicle-treated group. And the vehicle here, as we mentioned, it is the refined olive oil. So memory was improved in these mice. So this slide here, because I didn't want to go through many slides, but it summarizes the effect of oleuropein leaf extract spiked in olive oil. And we found that it was able to significantly reduce beta amyloid pathology, reduced in neuroinflammation, reduced oxidative stress using via different multiple mechanisms that are listed here, and also it improved memory function based on all these effects that were observed. And also it reduced the production of beta amyloid, which I didn't talk about it here. Now here, this is the overall effects of olive oil, EVOO, and its phenolic effect in the mouse brain. Enhanced BBB tightness and function, cerebrovascular function, reduced beta amyloid by different mechanisms, and also reduced tau hyperphosphorylation, reduced neuroinflammation, and enhanced antioxidant capacity, which collectively enhanced memory function. Now, it is always the question, okay, we see beautiful results in the mouse. It is working all the time. So the idea, the question here, is it going to work in humans? And for this study, we tested oleucanthal-rich extra virgin olive oil, and it was mainly the Governor brand that we used for this study. And we compared it to the refined olive oil, and this one we just bought it from the market in order to compare it. And to answer the question, does extra virgin olive oil prevent MCI is mild cognitive impairment, conversion to Alzheimer's disease. Because when we talk about Alzheimer's disease, there is always a step that precedes Alzheimer's that is known as mild cognitive impairment. With the idea here, we want to prevent its progression to Alzheimer's disease. So we have the subjects, we gave them extra virgin olive oil, we gave them the refined olive oil, and we over 6 months, we tested different parameters, and here I will present some of these parameters. So ROO for refined olive oil, and EVOO is for the extra virgin olive oil. And this results is from the MRI. We did MRI study at resting state, and we found that compared to the refined olive oil, the red color is the baseline, the green color after 6 months. So the refined olive oil didn't have any effect on the functional connectivity, no significant effect. However, with the extra virgin olive oil, we found that the extra virgin olive oil actually enhanced the functional connectivity. What does that mean? That the neurons from different regions are communicating with each other. And this is for sure, it is important for memory. Not only that, we found also, because I have interest in my lab about cerebrovascular function, the blood-brain barrier. So we did an experiment or the MRI here we did that it is the DCE, or the contrast enhancement study. We want to measure the permeability of the blood-brain barrier. So here with the refined olive oil, we found that there was no significant difference. It was the same. However, with the extra virgin olive oil, it enhanced the tightness or the integrity of the blood-brain barrier function, kind of restoring its function to normal level. Although the effect here was not significant, but we did additional study focusing on the hippocampus, and we found actually those with the extra virgin olive oil showed significant improvement in their cerebrovascular function compared to the baseline data, and it was significantly different. Now, the main question as outcome for us, it is memory. It is always about memory. So we tested we use different tests for the memory function, and here for the CDR sum of boxes, it is just a memory test. And we found, interestingly, that refined olive oil actually improved memory, and extra virgin olive oil also improved memory. So everyone will ask why refined olive oil improved memory. We should not ignore the effect of the good fats in olive oil, mainly which represents the highest percentage, which is the oleic acid. So we should not underestimate the effect of the good fat in olive oil, that for some reason, different mechanisms that we are establishing now, that it was able to enhance memory in these patients, like extra virgin olive oil. In addition, using another test, which is the vascular memory scale 4th edition anyway, so it just monitor the change in memory function, immediate, later, and whether they can recognize. And we found that we have larger number of subjects who were on extra virgin olive oil. Actually, they had better memory. But also, I will not ignore refined olive oil, because some subjects actually they had improved memory with their while being on the refined olive oil. So those are the results we got. Obviously, olive oil is good. Don't underestimate the refined olive oil, but for sure the phenolic content can enhance it. And this is here the acknowledgement for all people who worked with me, the collaborators, of course, and also for the funding that was, and also for those who provided us with the olive oil. And thank you very much.
Congratulations, Professor Kaddoumi. Thank you, Amal, so much. Ευχαριστούμε πάρα πολύ την καθηγήτρια Amal Kaddoumi για τα πολύ σπουδαία αποτελέσματα που ακούσαμε. Ξέρετε, είναι εντυπωσιακό το ότι οι ουσίες που υπάρχουν μέσα στο ελαιόλαδο μπορούν να φτάσουν μέσα στον εγκέφαλο είτε του πειραματοζώου είτε του ανθρώπου και εκεί να ασκήσουν μια τόσο ευεργετική δράση. Είναι πραγματικά κάτι το οποίο εμένα με συγκλονίζει όταν σκέφτομαι πώς ξεκινήσαμε πριν από 20 χρόνια να ασχολούμαστε με το θέμα αυτό χωρίς να ξέρουμε τίποτα για το τι συμβαίνει μέσα στον ανθρώπινο εγκέφαλο, και ξαφνικά βλέπουμε οι ουσίες του ελαιολάδου να φτάνουν εκεί και να έχουν μια δράση σε τόσο σημαντικά θέματα όπως είναι η λειτουργία των νευρώνων και η προστασία τους από την εκφύλιση. Η βελτίωση της μνήμης, η βελτίωση του ύπνου, πραγματικά πάρα πολύ σπουδαία ευρήματα. Ευχαριστούμε πάρα πολύ την καθηγήτρια Amal Kaddoumi για τα πολύ σπουδαία αποτελέσματα τα οποία μας παρουσίασε, τα οποία να τονίσουμε για μία ακόμα φορά ότι συμμετείχε ελληνικό ελαιόλαδο για την μελέτη αυτή. Σας ευχαριστούμε πάρα πολύ.